Savonlinna is in the middle of Lake Saimaa, which is the largest lake in Finland. Besides beautiful landscapes, Savonlinna is known for the opera festival held in an old castle. There is also a good amount of forest sector industry. For example, near to Luke, there is Andritz, which is a leading global supplier of systems for the pulp and paper industry. Luke's office and technological platform, including laboratory, are situated in the technology park together with Southeastern Finland University of Applied Sciences and companies of high technology. Welcome to Luke's technological platform in Savonlinna. Aseptic working is the base for in vitro research. For that, we have several hoods with horizontal flow. You can do microscoping and take pictures inside the hood. We have also two custom-made extra-wide hoods, which allows you to do group work or work with large set of material.
We study, for example, vegetative propagation of conifers and especially somatic embryogenesis of Norway's spruce.
We start the production of Norway's spruce somatic embryos from tissue which is 5 to 7 days from previous subculture. For research purposes, we scale the amount of tissue, place it to the tube filled with maturation liquid, shake a little, and pour the suspension to Büchner funnel filled with filter. The under pressure sucks the extra liquid away, and the filter with embryogenic tissue is placed on the maturation medium. Embryos are maturing in the dark in room temperature. When embryos are mature, they are moved to the cold room where the temperature is +2 degrees Celsius. Temperature stays steady in big room, and that allows the storing of embryos even over a year without decrease in germinability. When the production continues, embryos are germinated under LED lights. Embryogenic tissues and maturing embryos are kept in growing rooms, which have automatic control of temperature. Also, the cold room and germination rooms are controlled using the same system. There is sensors in every cell, and you can monitor the temperature from one place.
And over here we have the climate data from our growth rooms, and now you can see the temperature chart from the room 025. And as you can see, there's a clear difference between day and nighttime temperatures among the shelves in the growing room. And when we change the scale of the chart, we usually look at the chart between 15 and 25 degrees Celsius. It looks visually quite a lot different. Even though it is highly regulated chamber, we still have quite clear differences between shelves in the temperature when the lights are on, but when it's nighttime in the room and there's no growth lights, the temperatures among the shelves are quite close to each other.
Besides conifers, we study also broadleaf tree species. At the moment, Sakari is developing the method to cryostore and regrowth the elm trees. Elms are globally threatened by Dutch elm disease, and supplementary conservation methods are needed to protect their genetic diversity. Our facilities for cryopreservation includes the slow cooling device and room for over 30,000 samples. Automatic detection of oxygen level and emergency ventilation are important for safe working. Our big storage tank is automatically filled with liquid nitrogen. We have modern facilities to make genetic analysis, from DNA extraction to PCR analysis. We have studied, for example, telomeres of the trees using Western blotting. And now Jaanika is genotyping our Norway spruce SE lines with root rot resistance gene marker. Every week, we use hundreds of Petri dishes and glass jars filled with different kind of growth medium. Automation in filling the Petri dishes saves a lot of time. The system includes also the printer, which adds the name of the medium and the date to the side of Petri dish. With the custom-made hole in the wall and separate pump, we can utilize the large hood in the room next door. Aseptic work is based on clean laboratory and sterilization of the tools used in the hoods. We have invested in good design and modern devices, especially in the dish room. In all, we end users were able to participate in the designing of the building from the beginning, and we paid attention to ergonomy and easy and efficient use of the space. Thank you for watching and welcome to Savonlinna.